We have the next talk by Sahar. And she's a principal scientist at Bolivia Medical. It's a small company, a startup company from the University of Galway. And Sahar is also a patchliner user, relatively new patchliner user, and she will tell us about how ion channels involved in pain perception for arthritis. Yes, very much looking forward to your talk.
That's okay. I think so. Yeah. Thanks for the introduction, and I would also like to thank Sananium for giving me opportunity to talk today here. So yeah, I'm going to talk about knee osteoarthritis and see if biopolymer can be an answer and as a therapeutics. So what is knee osteoarthritis? So osteoarthritis is the most common form of knee osteoarthritis, which is mainly as a result of wear and tear and progressive loss of cartilage. And it occurs to people age of over 50 years, usually. Currently, there are 250 million people worldwide suffer from osteoarthritis, and patients consider pain as the most traumatic element of the disease. And 60% of patients describe their quality of life as poor due to the disease. So what are the current treatments? Well, up to 80% of the patients rely on oral pain medications, which are not long-term, they are short-term, they damages the organs, and they can be addictive in terms of opioids. There are also injectable treatments such as steroids, which again, they are short-term, 2 to 4 weeks, and it does damage cartilage. There are hyaluronic acid injectables, which have limited efficacy and pain relief. Stem cell and platelet therapy, which has poor efficacy, and eventually knee replacement surgery, which is highly invasive. It's very expensive, and patients still suffer from pain after the surgery. So there has been no improvement for the last 50 years on treatments for knee OA pain. So what is the emergent therapy? So peripheral analgesic targets a transient receptor potential 1, TRPV1 is got a fast track FDA approval for knee OA pain. So what is TRPV1? I think most of you know. So TRPV1 is a non-selective cation channels. It activates by a variety of signals such as heat, low pH, and agonists such as capsaicin, which is a natural compound from chili pepper. It brings calcium inside the cells, it initiates action potentials, and it's propagate into spinal cord and to brain as pain. So there are many companies been working on TRPV1 either as antagonists, so blocking TRPV1 and basically block the pain. But most of these companies actually failed their clinical trials because one of the disadvantages of using antagonists is patients develop hypothermia or hyperthermia, and mainly antagonists have a transient effect. There are also agonists. So agonists, as I mentioned, such as capsaicin, how it works, it will first synthesize the neurons, but then repeated applications of the agonist can actually desynthesize the cells and, of course, inhibits the action potential and give pain relief. So Centrexin, one of the big companies, they worked on capsaicin. They went up to phase 3, but they actually marginally failed the phase 3 trial. And we think the drug is working, but is not retained in the joint. And there is not much data out there, but we think they're using a bolus release of the capsaicin and they can't reach the efficacy needed. There are also Sorrento and Granatol. They're actually working on another natural compound from cactus, resin-aflatoxin, and they also are using the bolus release of RTX or resin-aflatoxin. So is targeting TRPV1 as a near perfect pain treatment? We think yes, and what Relivium is developing is a novel biopolymer, injectable biopolymer, which has a slow release system. So it can give more efficacy, it can give protections and lubrications. And we have optimized pharmacokinetics and pharmacodynamics of this biopolymer. And if we compare it to pipeline treatment, for example, such as regenerative drugs, so Merck has brought a product to clinical phase, but one of the things about regenerative drugs is that it does regenerate cartilage, but it has very poor efficacy. And another example, anti-nerve growth factor, which has actually rapidly progressed OA. It was actually halted by FDA in 2010, which then it was lifted in 2015. But big companies such as Pfizer and Eli Lilly brought products to clinical phase, but they actually failed because they couldn't reach the efficacy needed. So we think that RM010, what we're developing, it can give better efficacy and safety, and also the same if you compare it to commercial treatments such as oral analgesic and hyaluronic acid. So what is our pipeline clinical project plan? So we are using capsaicin as an example because it's a well-studied agonist. We use a polymer to make our biopolymer. And then we study kinetic release, mechanism of action, and cytotoxicity. And thanks to Nanion, we have used patch liner heavily to actually do all this study. And then we will move into our in vivo. We will start with a small rodent chemical-induced model in rat. And then we will move to OA patient canine study later. And we will do behavioral study to measure the efficacy and pain relief. So a little bit about chemistry. So as I said, we are using capsaicin as a model. So capsaicin is a hydrophobic molecule. And this is the structure of capsaicin in here. And this is the structure of TRPV1. So how it binds, capsaicin binds through this OH bind and this double O bind, and form hydrogen bonds to this S4 and S5 segments on TRPV1. And what we've done is we use selective chemistry to make a biopolymer which is soluble and is stabilized, and we have optimized the pharmacokinetics of that. So this is the proton NMR of biopolymer. So this is the proof purified solid state of our small molecule, which is capsaicin. So these are aromatic protons, which are these ones here, and aliphatic protons, which are here. And these peaks are actually the polymer. So this shows a successful integration of small molecule capsaicin into our polymer. So this is only a fraction of chemistry I'm showing here. There's a lot more to it. Then for in vitro, so first we had to choose a cell model, and we had quite a story about our cell model. I just want to put you through it. So we bought this HEC-3B cell line from a very well company. They were RFP positive, so we can easily quantify them. We measure with patch liner. They have very specific temperature response. We measure them in calcium imaging. They have response to capsaicin and reduction in the presence of antagonists. But when we wanted to do the patch clamping to measure the inward current, we couldn't see. There was no response to capsaicin. Probably less than 1% of the times we would see a response. So we went through a lot of troubleshooting, and one of the first things we thought, okay, maybe the cell line is not purified enough. So we went through a lot of single cell purifications of the cell line. So we created actually a more purified of HEC-3B1, still no response. We actually doubted our capsaicin. So actually, thanks to our chemist, we went and did NMR on capsaicin. Actually, the capsaicin that we bought from a very well-known company, it turns out it was not capsaicin. It was labeled as capsaicin, but it was not. And this is something probably we've been using in the lab, and probably most of people has been using in the lab. It was a very close agonist as well, but it was not capsaicin. So we bought a correct capsaicin, still no response. We actually sent ourselves to Nanion because we told them we may be doing something wrong. Nadine kindly tested the same, no response. So we kind of gave up at this point, and we couldn't really find out why we had temperature responses. One of the reasons is that the segment that binds to temperature, activates temperature or capsaicin is different, or the way that TRPV1 is expressed with the transfection was not accessible to the capsaicin. What we didn't investigate, this took us probably 4 or 5 months, and it really delayed our project. So we moved on, and we went and bought another cell line, the CHO human TRPV1 from BSES, and of course, magic happened, and we had our nice DR curve. Even though we had a nice DR curve, but after that we went through a lot of other troubleshooting. I'm not gonna explain now, but we were confident at the end that the result we were getting was good, so we were moved on. So then we did the kinetic release study to look at the release of the capsaicin from biopolymer. So just to explain here, I'm showing here the percentage of capsaicin release. So this is based on the maximal induced current that we've done all of our results on the patch liner, and then we put the unknown concentration on a DR curve, and then we calculate the percentage of capsaicin release. So there are many things that I'm showing few of them here that it was important for us to investigate from our biopolymer. So first, we need to know that our biopolymers are stable, because this is important for shelf life of our drug substance. So we want minimal release at 4 degrees. So in here you have the red and burgundy color here, and we can see at 4 degrees they are very stable and they are very slow release. Also the capsaicin content. So the capsaicin content in the biopolymer also affect the release of capsaicin from the biopolymer. So in here we have blue, which is a high capsaicin content, versus black, which is a low capsaicin, and we can see that there is the release kinetics is different. The solvent we actually dissolve our biopolymer actually affects the release, and this is very important because we need to maintain the stability of the biopolymer, not only at the 4 degrees when we keep the drug, but also at 37, because this is our body temperature and this is where we're going to inject the biopolymer. So you can see in the black line, we have a very nice slow release, but if we do solvent B and C, we have a very sharp release, and this is something that we don't want, of course. And also the choice of polymer is also important. So different polymer can give different release of capsaicin. So these are only few of the parameters I'm showing. There is a lot more to it I'm not talking about, but these are a few of them that we study for the kinetic release. So next, we want to understand the mechanism of action, but not just on the CHO cell line, but probably to a more DRG cell line. So we have chose ND723, so it's a hybrid cell line, is a DRG cell line. We have transfected it with human TRPV1, and we started doing some functional assays. So at calcium imaging, it responds to capsaicin, and on the patch liner, we have done DR curve, and we have very high efficiency, over 90% efficiency of the TRPV1. So this is important for us because we want to understand the acute and the chronic effect of capsaicin from the biopolymer, not on just the TRPV1, of course, on the NAV channels. And these ND7, they have NAV 1.7, which is important for us. So we've done some preliminary results. So this is just the addition of capsaicin. So this is when the cells become synthesized, so we can see increased current in the presence of capsaicin. If we pre-treat the cell, we can see that there's inhibitions. And then in here, we have a cumulative effect of capsaicin with reduced sodium current. Again, this is important for us to see the slow release of capsaicin effect from the biopolymer on sodium current inhibitions, on action potential firing, and we would like to find sweet spot which is needed for efficacy. So in summary, we have used as a selective chemistry to form a soluble TRPV1 biopolymer, which has optimized pharmacokinetics and pharmacodynamics for sustained release, that we can selectively ablate the nociceptors. We have reliable cell models, so we have CHO and ND7, but we are hoping to move on to IPC-derived DRG neurons later. And we have established rapid kinetic release and we are studying the mechanism of actions with the help of patch liner. So what is the promise of RM010? We are hoping that patients move from having damaging oral pain medications and poor pain relief and not having good mobility and poor quality of life to patients regaining their mobility, and they have a pain-free life and they have a better quality of life. I just like to thank this all work is done with amazing things. So Relivium is Alison and Barry are the co-founders of the Relivium. Manaz here is in charge of the in vivo. Of course, this project is in the collaboration with University of Galway in physiology mainly with my former PI, Leo Quinlan. And we have 3 chemists. So Christoph is the senior chemist, which is hiding behind all of us in there. Hara and Yahira, they are the most of the in vitro work here, which they are in the audience here. And people in pharmacology and chemistry. Last but not least, Nanion, and special thanks to Nadine, which has been super helpful. And I don't think we could have been here without her. And Christian for all the technical help for the last few months. And thank you all for your attention. Thank you.
Thank you, Sahar, for a wonderful talk.
Thank you.
Questions from the audience?
Okay, one question. So have you thought about how much you have to inject into the knee? Do you have any idea? Because this is one of the
we know from the literature and what they've done from, for example, Centraxion and other companies. So if you inject a lot of agonists, which they've done, you can first, it's not retaining the joint and it can come with some side effects. So we're trying to study the mechanism of action in vitro, and so high amount is not good, low amount it's very weak analgesic. So that's why we want to find the sweet spot in here. So we have some idea, but we need to do some in vitro mechanism of actions for the slow release and a cumulative effect.
Not referring to compound concentration, but to volume.
Oh, the volume.
Yeah.
So in animal, we do a 50 microliter.
Okay.
In human is a 3 ml volume, because that's a 1-to-1 ratio with the volume of synovial fluid in the jaw, in the knee.
Okay, thanks.
All right.
Not really to your technique, but it's more like conceptual here, but actually also to the choice of the compound that you want to administer there to deliver to the place. So first conceptual part is, yeah, you are clearly addressing the symptom. You want to numb the pain to make the people move more, but would not that promote enhanced bone destruction, actually? That's one thing. And second thing, I have this issue with the choice of the agent, the capsaicin. Would not it stimulate actually inflammation more? Would not it be better to use some other compound which would not be associated with inflammation, with enhancement of inflammation? Some kind of antagonist. Because capsaicin would overload TRPV1 and it will kind of numb it. It goes like to the plateau and then it shuts down at the nerve level. But would it not be better to shut down activity of the channel to prevent the signal from appearing, which also would have another benefit of not actually inducing inflammation, extra inflammation?
Well, your second question first. I forgot the first question already. Well, I think I said there's loads of companies use antagonists, and there is a problem with hypothermia and hyperthermia. So they went to clinical phase 2 and 3, and they all failed to actually achieve the efficacy. So it's not, I think that's simple. And in terms of capsaicin, okay, we're not going to inject. So capsaicin we use as a model. It's not our final agonist that we're going to use. And the thing is, we're not going to use capsaicin on its own into the joint. We put it in the polymer. So we have a biopolymer. The release is very slow. And actually, we have some preliminary data that it can actually protect the inflammation. That can actually reduce the inflammation in the joint. And the main reason is because we're using the polymer that we're using with the capsaicin, yes. No, so we were testing different polymer to see which one is the best, but we have one polymer. Yes, we are doing that. Yes, yes, yeah. We are studying, but I'm not showing all the data here. So yes, yeah.
There's a question online.
Read it as well, actually. But Eleanor is asking, can you comment on prolonged usage of the drug? For example, for people younger than 50 expected to take it for decades.
I don't know the answer for that. So prolonged exposure. So, I mean, what we are, maybe if I'm answering correctly, so we are hoping that this biopolymer, we can give one injections and it can give 9 months pain relief for patients. Currently, I think the Centrexin, what they were trying to develop, and Sorrento and Grantel, they are hoping to do a 6 months, but of course they come in with side effects. So they need patients need pre-treatment and after treatment in the clinics when they have the injections. For younger generation, I don't know the answer for that, no.
Thanks again. I think we move on to the next speaker.